In this video, we're going to take a look at what enzymes do, and at the differences between the lock and key model versus the induced fit model of enzyme action. Then, in the next video, we'll take a look at the conditions that can affect how enzymes function. In order for a living cell to function properly, it has to carry out a huge number of chemical reactions every day. One of the problems with this, though, is that most chemical reactions are naturally quite slow. One way to speed up reactions is by increasing the temperature. But this runs into a few problems. One, it can require a lot of energy to keep an entire organism very warm. Two, high temperatures can damage our cells. And three, higher temperatures will also speed up all of those non-useful reactions that we don't actually want to take place. A better way to increase the speed of a chemical reaction is with a catalyst. And we can define a catalyst as a substance which increases the speed of a reaction without being changed or used up in the process. This last bit of the definition is really important because it means that catalysts can be used over and over again without being used up. Now, you might remember from chemistry that catalyst is a general term, but an enzyme is a particular type of catalyst that is made by living organisms, and so we sometimes call them biological catalysts. Enzymes are basically just large proteins, so like all proteins, they're made up of long chains of amino acids. But once these chains of amino acids are folded into more complex shapes, they create unique enzymes that can catalyze, which just means speed up, a particular chemical reaction. So now that we know what enzymes are, we need to look at how they work. If you think about chemical reactions, they often involve taking one thing, which in biology we call a substrate, and breaking it into multiple parts, which we call products. Or it could be the reverse of this, where multiple substrates are combined to make a single product. Either way, all the enzyme does is speed up the process. They're able to do this because they have an active site, which is a region on the enzyme with a unique shape that's specific to the substrates of the reaction. And this point about the shape being unique is really important, because if the substrate doesn't fit the active site, then the reaction won't be catalyzed. This feature allows enzymes to be really specific about the reactions that they speed up. There are two models of enzyme action that you need to know about: the original lock and key model, and the more realistic induced fit model. At first, scientists thought that the substrate had to fit perfectly into the active site, just like a key fits perfectly into a lock. However, we now know that the enzyme actually changes shape slightly, like this, so that it fits more neatly around the substrate. This idea that the active site changes slightly to perfectly fit the substrate is known as the induced fit model. That's it for today. If you enjoyed it, do give us a like and subscribe, and hopefully we'll see you next time.